Hey guys, so this video is going to be over phylogenies, which if you want to think of it initially, you can kind of picture it like a family tree. I will go ahead and tell you now that this particular topic is one that you will learn and understand much better by doing and making cladograms and phylogenetic trees than by the notes that we're about to take. But we're going to step through and talk about some of the important vocabulary and how you should interpret certain images and diagrams, and then in class we'll do a bunch of practice over it. So here we go. So let's start off by talking about taxonomy, which does tie into phylogenies, cladograms, phylogenetic trees. Classification, or the method of sorting organisms or placing organisms into categories, was originated with a scientist called Carolus Linnaeus, and that happened in about the 18th century. So it's been around for a long time. It's all based on structural, both inward and outward similarities. So we're sorting them based on how they're structurally similar or how they're structurally different. And we can get those similarities based on how they're made up, what we can physically see of the outside of the organism, but we could also take into consideration internal things like DNA and protein structures on the inside. It's a hierarchical scheme, which just means that it goes from small to big, large to small. The largest, most inclusive group is the kingdom level. That was original to Linnaeus's early theory. In more modern times, we have replaced the largest grouping with the term domain. So the domain is now the most inclusive group, and then after that comes kingdom and phylum and order and class and genus and all that good stuff. The most specific grouping, so the one that has the least amount of organisms in it, would be the species. So we start off as a kingdom, we start off in a domain, and then we trickle down to the species level. One of the things that biologists do is that organisms are given a scientific name. The scientific name is Latin and is composed of two parts of this taxonomical structure, the genus followed by the species name. So here's an example of the scientific name for a cheetah. This first part, the one that's capitalized, would be the genus name, and then the second part, the one that's not capitalized, would be the species name. So taxonomy is pretty much the classification of an organism based on shared characteristics. What are the things that it has in common with other organisms? So I told you a couple of slides ago that in later times, kingdom was the biggest grouping that we had. A more recent development is the category of domains, which was proposed by this guy, Carl Woerth, and he did so based on RNA differences between prokaryotes and eukaryotes. So pretty much these are the three domains that all life fall into. The prokaryotes are divided into two groups, bacteria and archaea, and then everything that's not a prokaryote belongs to the domain eukarya. The organisms are grouped again from species, but this time to domain, and as we go up, the groupings are more inclusive. So at the species level, we only have a couple of organisms because you have to have many similarities. But as we go up into the kingdom and the domain, you get way more organisms because there are not as many things that you have to have in common. So the taxonomic groups from broad to narrow start with domain. So the domain has the most organisms, followed by kingdom, phylum, class, order, family, genus, and then finally the most precise or specific would be species. In this hierarchical system, each taxonomic unit is called a taxon. And classifying organisms according to their shared characteristics also gives an indication as to their evolutionary history. So that's why taxonomy and phylogeny kind of tie in together. So I've thrown the term phylogeny around a bit, so let's talk about what that means. Phylogeny is a study, it's a science actually, of the evolutionary relationships among a group of organisms. From this study, we can generate a diagram that we can call a phylogenetic tree. So it's a tree-like structure which illustrates or maps out the evolutionary relationships of a group of organisms. It can be based on several things. One of the things that we look at would be morphology, so actual shape and structure, and we also look at the fossil record. So that's how we compare extant versus extinct species, things that have already died compared to things that are currently and still living. We can also look at embryology, the development of a fetus into an actual organism. And then we can also look at DNA and RNA and protein similarities. So all of these things can be used to build a phylogenetic tree or to base a phylogenetic tree off of. So there are two things that we can use to illustrate phylogenies in biology. And a lot of biologists use the following terms interchangeably, but there really is a difference between the two. We can use cladograms to illustrate phylogenies, but we can also use phylogenetic trees to illustrate phylogenies. The difference between the two is one is a hypothesis and the other is the one that represents true evolutionary history. So a cladogram is what we use to represent a hypothesis about the evolutionary relationship or history of a group of organisms. This is how we think they fit together. This is how we think they have evolved based on who is in this group. Whereas a phylogenetic tree is one that represents the true evolutionary history of the organism. So we know for a fact, or we're very, very certain that this is the actual truth. So this one's a hypothesis, where this one's more proven, if you want to think of it this way. So really quick, I want to go over this part of the slide. Phylogenetic trees don't really look like trees. They just look like a bunch of lines that kind of split off and diverge at different places. But the line itself represents a species or a population. And as the line progresses, it represents how that population changes or progresses throughout time or a particular lineage. Wherever it splits, like it's doing in this part of the picture, the point where it splits is called the node, also known as the branching point. And a split just shows where the lineage splits into two descendant lineages, so two different populations, if you will, evolve independently of one another. All right, so here's an example of a phylogenetic tree. This one is based on the organisms that are closely related to our modern day elephants. And as you can see, it doesn't look like a true tree, but it does have all of these branchings, which is where we get that tree comparison from. The length of time that they run for is normally represented in millions of years. Sometimes it's billions of years. It just depends on the organism that we're talking about. Any organisms that make it all the way to the top, like the hyraxes over here and manatees, and then these different kinds of elephants, Asian and the two kinds of African elephants, that says that these organisms are still extant. They are still living. Whereas organisms that haven't quite made it to the top, like these guys here, that tells you that these are extinct. They have died out before present day. Okay, so just so you understand why some of them stop short and others go all the way through. So this is almost like reading a graph, because if you look at it, you can see that where some of this branching and this splitting took place actually corresponds with a timeline. So, you know, like right here, let me find a decent one. Like if we look at this guy right here, it looks like this split took place about 34 million years ago. So just so you understand what that means. What else do I want to tell you? This one would be a phylogenetic tree because this is not a hypothesis. We know, we're fairly certain, almost 100% certain, that this is the evolutionary history of these particular organisms. So here's an up close of a smaller one of these trees are cladograms. And let's just talk about some terminology. So right here, where it says taxon A, B, C, D, etc., this is where we would place the names of the organism if we were working with individuals, or it's where we could place the name of that taxonomic group, so kingdoms or species or genus or however far within the taxonomic group we're working to. So another definition for a taxon would be a group of species that's designated by a name. So kingdom, class, phylum, order, genus, species, etc. There is a term called sister taxa, which is illustrated by these two right here that I'm boxing off for you. And sister taxa are groups of organisms that share an immediate common ancestor. So if we follow their lineage from this one and then that one, you can see that they both branch off from the same point right here. And they would have shared common ancestry via whatever organism this originally was. Okay, so technically every node, every branch point should give rise to two separate lineages. So if we look at this top one, here we can see that we branch off into this guy, and then we branch off into something else that eventually branches off into something else. But every node point gives rise to two different lineages. When you have a situation where you have three different lineages coming off of one branch point or one node, like we have here with 1, 2, and 3, then we have a situation called a polytomy. So a polytomy is what happens if more than two lineages are shown off of one branch point. And this just tells us that there is an unresolved pattern of divergence. We're not really certain who came from whom. So, in other words, this is still a hypothesis at work. This would be a cladogram. Okay, so be aware that these trees can be flipped around, like the order of the organisms can be rotated. So if you look at tree A versus tree B, you would see that the order of the organisms or the taxon groups are flipped around, but that doesn't change the evolutionary relationship. That remains the same, even if we move around to a certain degree where those groups or names are found. So another term that I want you to learn is the term clade. So technically a clade is any taxon that consists of all of the evolutionary descendants of a common ancestor. So in this picture, all of these colored rectangles would represent different clades. So this yellow rectangle represents every single evolutionary descendant of this particular common ancestor. This blue one, notice that it includes the yellow rectangle now. This would include all the descendants of this common ancestor. This red one, all the descendants of this common ancestor. And then the green one, all the descendants of that common ancestor. So I'm going to mark those out. So this would be where your common ancestor is. Common ancestor. Common ancestor. Common ancestor. And realize that it is possible for multiple organisms to be part of multiple clades. Let me rephrase that. Realize that it's possible for one organism to be part of multiple clades, just depending on which common ancestor we're basing it off of. So another term for clade is the term monophyletic group. A true clade, or a monophyletic group, is one that contains a common ancestor and then all of its descendants. So if we pay attention to picture A, this would be the common ancestor that we're going based on. And then if you look, you'd have a descendant here, so that's descendant number 1, and then the lineage splits again into two separate descendants, but they all originate from whatever or whoever this is right there. This would be a monophyletic group or a true clade. Everyone here is connected or related somehow to this originator down at the bottom. So that would be a monophyletic group one common ancestor, all related. Another term is paraphyletic group. So if we look at picture B, that shows a paraphyletic group, and it is one that has a common ancestor, but does not contain all of the descendants. So if you look, here's our common ancestor, and then we have family grouping off here and here for 1, and then here would be 2. And then on this side, we branch off and we form whatever this is over here, which is 3. But note, I'm going to do this one in black, that even though G does branch off of the same and common ancestor, it's not included within the group. So this would be what we call a paraphyletic group. We have a common ancestor, but not all of the descendants are included. Okay, the group doesn't contain everyone that has lineage from this common ancestor. The last one is called a polyphyletic group. And in a polyphyletic group, all of the organisms do not have a unique common ancestor. So all the descendants didn't originate from the same common ancestor. So here's a common ancestor there, which is fine. But if you look, we split off into these 2 groups and then these 2 groups, which is also fine. But we've also included this guy here in this grouping. Well, he does not share lineage with them. This is their common ancestor, but this is not. This common ancestor here does not belong to him. He does not share lineage with the common ancestor of the other 4. So this would be a polyphyletic group, just meaning that these descendants originated from multiple ancestors or organisms. So now we're going to tie in our phylogeny study with a little bit of evolution. And I want to introduce the terms Anagenesis and Cladogenesis. So let's start with Anagenesis. Anagenesis is also known as phyletic change, and it's the accumulation of a bunch of different changes in one species that leads to the complete change of that species over time. So think of it as the evolution of an entire population. To put it in layman's terms, you have one kind of organism. Over 50 years or 100 years or 2,000 years, however many years, they change into a completely different species or a different population, and the original population no longer exists. So after a certain number of changes have accumulated, the ancestral or original population is considered extinct, and whatever the new organism is the going population. Okay, so we literally go from having one kind of bird, like if we use this picture, to a bunch of changes taking place, and now we have a completely different species of bird. Note, based on the picture, that the 2 don't exist at the same time. And by the way, this brown arrow just represents time. Okay, so that would be anagenesis. Over time, we have enough changes in our original species so that we have a completely different species, and the 2 don't exist at the same time. Okay, the other term was cladogenesis. And in cladogenesis, what happens is you have a new species that develops as a result of changes, but the original species is remained. So it's the budding off of one or sometimes even more new species from a species that continues to exist. Because if you look at the picture, here is our original bird. As time passes, we still have our original bird, but we've had enough changes to part of this population that a new species has formed. Okay? Note that both of them exist at the same time. So with cladogenesis especially, it normally involves some kind of physical separation, and that could be a mountain, a lake, an ocean. Something is physically preventing them from being in very close contact with each other, and that allows them to evolve separate to each other. So they become 2 different species. So for a long time, people thought that there was no evidence to support evolution. We had a proposal that it happened through natural selection. That was courtesy of Charles Darwin. But people thought, there's no concrete proof. You have no evidence. What are you showing me that tells me that this evolution thing really does happen? Ironically enough, our first bit of evidence is found in the ground beneath our feet in the form of the fossil record. So the fossil record serves as our first piece of evidence that evolution really does happen and it does occur. Unfortunately, not everything is capable of being fossilized. You have to have hard parts, bones or shells, something that won't be destroyed easily over time. Even if you do have those hard parts, sometimes the environment is not conducive to fossilization. So it means that our fossil record is incomplete. While it does provide evidence for a lot of organisms that are no longer around, while it does show that at some point in time they were with us, there are also a lot of organisms that just did not have the parts or the environment was not conducive to them forming a fossil, and they're not a part of the fossil record. So this picture really quick showcases how fossils get embedded within soil layers and how the oldest fossils are always found in the bottommost layers of soil. So you can kind of tell the age of a fossil based on the rock content and what's around it. So younger fossils would be found closer to the top and older fossils would be found closer to the bottom. So these guys would be more ancient fossils, whereas these guys would be more recent fossils. Another bit of evidence is homologous features, just structural features that organisms have. Now, even when cladogenesis takes place and one group splits into two distinct groups that are evolving independent of each other, they keep a lot of the features of their common ancestor. So any feature that's shared by two or more species and is an inherited feature from a common ancestor are what we call homologous. Homologous features can be things that are inherited, such as anatomical structures like bones within your forelimb, or DNA sequences, or proteins that all of those species have in common. Right, so here's a picture that I'm sure you've seen a couple of times. It shows the forelimbs of a whale, a bird, a dog, and a human, which at first glance probably don't look like they're very much related. But it shows the fact that, see how these bones are color-coded? Each color represents one kind of bone. So note that every organism, every forelimb has the same bones in pretty much the same place. Now the structure might have changed a little bit. It's just an adaptation to whatever environment that organism currently lives in, but they're still present and they're pretty much used for the same thing. So during the course of evolution, traits do change, but the original shared trait is always called the ancestral trait. If this trait is found in a newly evolved organism, it is called a derived trait. So it tells you that this particular feature comes from the common ancestor of the organism you're looking at. Any feature that's shared by two or more species is, again, as I said before, called homologous. So it's called a homologous structure or a homologous feature. So just know in terms of ancestral traits, those are the traits that came from the original ancestor, so the older trait or the older organism, whereas derived traits would be traits that you see in newly evolved organisms, and they came from that common ancestor. They were passed on in the genetic makeup. Now, it is possible to have structures that are similar in terms of what they look like, maybe even in terms of what they do, but they're not an inherited feature or trait. So those kinds of structures are called analogous structures. So again, analogous structures are those that are similar in structure, but they're not inherited from a common ancestor. So a good example would be the wings of a bird versus the wings of a bat. The bones in the wings of birds and bats are homologous. They have the same pieces, humerus, radius, and ulnar, finger bones, thumb, wrist bones, etc. But the wing structure did not evolve from the same ancestor. So then you're probably wondering, well, if they don't share a common ancestor, how come the wings look so similar and how come they're used for the same thing? Well, that has to do with the similarities in environment. Both of these organisms fly, okay? So the physics necessary for flight is going to be what allows that structure to develop. That is going to be the selective pressure. That's what natural selection is acting on, and that is what is responsible for the similar shape of the wings. So if you even think of like an airplane's wings, similar shape, used for the same purpose, but definitely not an inherited trait, okay? It's very important for you to understand that while we can use homologous structures to develop and establish a phylogeny, we never, ever use an analogous structure to develop a phylogeny. Okay, so that should never be a basis for how something is evolutionarily related. All right, so we're going to quickly talk a little bit more about analogous structures, and I'm going to give you a few more examples. Analogous structures really do evolve because of similar selective pressures. Those organisms exist in similar environments. They come up with similar issues and situations, so they evolve similarly to each other. When this kind of evolution takes place, it's called convergent evolution. It's where both organisms, even though they're not related, they may not even be living in the same place, like country or anything like that, but because their environment is so similar, they're exposed to the same conditions and they evolve similar ways of coping with their situations. So here's an example of a mole and a marsupial. So this is a marsupial mole and this is a placental mole. Note that they both have the name mole. This guy's found in Australia and New Zealand. This guy is found in Europe and America. So if you look at just their overall body shape, it's about the same. Notice that they both have really large forelimbs, huge claws, sharp-looking claws, almost like they can dig with them, which is exactly what they do. And they have a fairly prominent snout on both, which they use as a sensory organ. Never met, will never meet, nowhere close to each other, but they evolved the same body type, the same claws and facial features because they live in pretty much the same types of environments, just in two different locations. One in Australia, one in Europe and America. Okay, another reason that analogous structures can exist is due to what we call evolutionary reversals, where organisms kind of go back, if you will. So when we think of how life evolved, we know that fish existed long before animals that walked on land. Animals that walked on land are called tetrapods. But an example of an evolutionary reversal would be cetaceans, which are things like whales and dolphins. Originally, whales and dolphins were tetrapods. They lived on land, they had 4 limbs, but they reversed. They went through evolutionary reversal, went back into the ocean, and adapted to a new lifestyle based on their environment. So when you look at a whale or a dolphin, they have a very fish-like appearance. Long, streamlined body, huge tail fluke, they have flippers instead of legs and claws and all of that. But if you could dissect them, you would realize that they still have the same 4 limbs that we do. Even whales do. Theirs are just so small, they're hid within their pelvis and you can't actually see them on the outside of their body. They look a lot like fish. Just general body structure, they look a lot like fish. But that's because they live in the same environment that fish live in. So they have to be able to cope in that environment. So now let's talk about some DNA and some proteins. So homologous structures are coded by genes that have a common origin. These genes can mutate, and sometimes they do, but they still retain some or most of the ancestral DNA that originally gave rise to them. Through genomic sequencing and computer software and a field of biology called systematics, we are able to identify what these genes are. So we call these genes molecular homologies. Molecular because they're DNA-based, and homologies because they're similar from the original common ancestor all the way through its descendants. The more closely related two organisms are, the more DNA sequences they will have in common, and the more those sequences will be alike. So to give you a visual to go with what we're talking about, this would be an example of a DNA sequence. And the colored blocks here and here would represent DNA homology. So these are from two different organisms, but you would see that kind of the same bases exist in both organisms. So it just tells you that these guys have some similarities. They have some evolutionary history. So the identification of these DNA homologies led to the term being coined a molecular clock. And the hypothesis goes that among closely related species, a given gene usually evolves at a reasonably constant rate. These mutations can be used to kind of predict times of evolutionary divergence. So pretty much we know how often you see these particular DNA sequences, and when we don't see them or when the sequences change, it suggests that some kind of divergence, some kind of evolution has taken place. All right, so this graph just showcases the timeline for the replacement of a particular gene. In this case, we're looking at the amino acid replacement of hemoglobin, and we find hemoglobin in anything that has a red blood cell. It just shows that this occurs at a fairly constant rate over about 5 million years. The slope of the line represents the average rate of change in the sequence of that particular molecular clock. So it just kind of backs up that these genes tend to evolve or mutate at a specific time period. And every time you see these little like yellow data points, that suggests that a replacement took place or a mutation took place and that divergence has occurred. Now be aware that different genes are going to evolve at different rates, and there are a lot of other factors that can affect the rate at which a gene will evolve or change. Okay, so this picture is just showcasing a particular gene, and this part of the diagram shows the common ancestor. So note that after about 25 million years, we have two descendant lineages that have diverged off of this common ancestor. We have one that's going this way and one that's going that way. Note that each lineage had a single base pair change. So a single mutation took place, and now these guys differ from the original ancestor by two bases. So there's a difference here and a difference here, and they both differ from this common ancestor. So we keep evolving, or we keep going, and after about 50 million years, note that, again, we still have our two descendant lineages, but they've diverged even further by another base mutation, and now they're different from each other by 4 bases. So differences here and here, especially compared to their common ancestor. So it's just showing that as time progresses, this particular gene is changing. It's not changing very quickly, but it is changing. So molecular clocks have now been applied to some of our more common and everyday, but yet still advanced biological questions. We can use them to study genomes that change fairly quickly, like those of viruses. So in particular, HIV comes to mind, which is a retrovirus. Using a molecular clock, it has been estimated that the HIV virus entered the human population in the 1960s, but that the origin of the virus dates back to the 1930s. And again, they're just looking at how quickly those mutations are happening and what year they're happening in. Now, is this a precise science? No. If you notice, all of our data plots kind of cluster around this like 1990 to 2000 area of the, or not 1990, but 82, 83 to 2000 area of the graph. So you kind of use a best fit line and average from there. You adjust it as necessary. So especially now in modern times, we use molecular clocks a lot to clean up debates that we're having about phylogenies, particularly ones that we we can't figure out just using like anatomical structures and things like that. We look to the DNA to lead us. So if you look at this diagram down below, it shows 3 species, the species A, B, and C, and then their common ancestor. So note that species, and by the way, these little bubbles represent mutations. So note that species A and B both differ 4 mutation points from their common ancestor. So 1, 2, 3, 4, and 1, 2, 3, 4. Note that species C also differs 4 mutation points from the common ancestor, 1, 2, 3, 4. Note also that species A and B differ along 4 points of mutations from each other, but they each differ 8 points of mutations away from species C. So it suggests that species C split, the lineage of species C, split almost twice as long as the lineage of species A and species B. So these could literally be 3 separate organismal species, like, you know, a cat or a dog or whatever, but it could also be one strain of a virus and all of the different mutations that it has done in the past few years. So molecular clocks have become invaluable tools, especially as it relates to phylogeny. All right, so how do we make these phylogenies? How do we make these trees and cladograms? Well, there are 2 rules that you always have to follow and take into consideration. The first rule is called the rule of maximum likelihood, and the second one is called the rule of maximum parsimony. Maximum likelihood states that whenever you're thinking of multiple hypotheses for a phylogenetic tree or for a cladogram, you should take into account the one that reflects the most likely sequence of evolutionary events. And you do that based on what you know about DNA changes over time. The second rule states that, that's the one that's about maximum parsimony, states that when you're considering your multiple hypotheses or explanations for your observation, you should first investigate the simplest explanation that is still consistent with the facts. So don't go all hardcore and like beast mode and just try to make it more difficult than it is. A lot of times with these phylogenies, the simplest method is normally the one that works out to be evolutionary correct. So when it comes to constructing or reconstructing a phylogeny, there are a couple of ways that this can happen. It can be given to you in a table format that looks like this part of your diagram, where you'll know how many differences different organisms have when compared to each other. And this could be a percentage. Sometimes it's just a number of sequences. They can use different tables, and I'll show you a couple of various ones so that you can get comfortable with them. So you always need to take that table into consideration if you're given one. Sometimes you're just given a group of organisms. Sometimes it's the physical thing, or it's a picture that represents that organism. And then you use anatomical features to build your phylogeny or your cladogram off of. Whenever you're given a table that has numbers, those numbers are going to represent differences between gene sequences. So to read the table, we have human, mushroom, tulip, human, mushroom, tulip. So between human and human, there are no differences, and we expect that because they're the same organism. Between human and mushroom, there are 30% gene differences. Okay? Between human and tulip, we're now looking at 40% gene differences. And of course, between mushroom and mushroom, there are no differences. Between mushroom and tulip, there are 40% differences. And then between tulip and tulip, there are no differences. So that's how you would read this table right here. Now, based on these numbers, 2 different cladograms can be constructed. And you're going to look at the sum of the percentages from where you think they diverge to create your cladograms. So let's look at our 2 options. So if we look at tree number 1, here's where our common ancestor would exist, who or whatever that is. And then we would start moving up the tree. So here there is a 5% difference, or there's 5% here. We'll talk about what that 5% means in a little bit. and then 15%, and then 15%. So we know that between a human and a mushroom, there is a 30% difference. So here's where that 30 comes in. We have 15% differences here, and another 15% differences here. 15 plus 15 is 30. So that tells us that these two evolve fairly close to each other. Now, why would we not take into consideration that 5%? Well, because they're both, up until this point, they're both sharing the same genes. This 5% is something that they both contain. Okay, so hopefully I made that clear. We know that the tulip has a 40% difference between there's a 40% difference, between the human and the tulip. So those numbers would come from the 15 here, and the 5 here, and the 20 here. So why do we now count the 5? Well, look at where the branching took place. Here was our common ancestor. We have our first branching point right here. And then we go this way to form our human, and we go that way to form our tulip. So this 5% that the human has, the tulip doesn't have, that would be 5% base sequence difference between these two. So hopefully that makes sense. So another way to read this would be the bigger the differences between, the further apart they should be on your tree. So you end up with a tree that shows the human, and then the mushroom, and then the tulip. Now, this other tree that we could generate, this would be tree number 2. Sorry guys, my laser does not want to cooperate. So here we go. Here's tree number 2. And it shows our common ancestor here. Sorry about that. I'm tired. And then we have our mushroom, our person, and our tulip. So if you look at the numbers, the differences still need to add up to what it shows on the table. We still need to have 30% difference between our mushroom and our person. So here we have our 15% and our 10%, and then our 5% to give us our 30. We would take the 10% into consideration because don't forget, it splits right here. And we go this way for the mushroom and that way for the human. So this 10% difference here is something that our mushroom would not have. For our tulip, there should be a 40% difference. And we would get that 40% by the 25% difference here, our 10% difference here, and our 5% difference here. Again, we're counting the 10% for the very same reason we counted it the first time. The fact that it splits, and this 10% is something that this mushroom would not have in terms of those sequences. So at the end of the day, the table says that we have mushroom, then human, then our flower, our tulip. But out of these two, the one that makes the most evolutionary and biological sense would be table 1. So this is the one we would choose. We got 2 tables based on the data, but the one that makes most logical evolutionary sense is the simpler of the two, this table. When we work on our phylogeny lab, you'll see this in a better light. All right, so like I mentioned, sometimes instead of percentages, you just get numbers. So this is what a table that represents that would look like, and this is how you would make a cladogram based on traits. So step 1 is always read the table, examine the data that you're given, and then you propose a cladogram based on what you get out of the table. So all these numbers just represent how many similarities and differences we have between the two. So and we're looking at the following structures: a backbone, a jaw, 4 walking legs, an egg with a shell, and hair. And we are comparing a lancet, a lamprey, tuna, salamander, turtle, and leopard. So if they have those things, they get a 1. If they don't have those things, they get a 0. So as you can see, our lancet is a very primitive organism. It doesn't have any of the structures we're looking at, whereas our leopard would be our most derived organism because it has all of them. It has evolved all of these things. So we would call this lancet a outgroup, and that is the group that is closely related to the organisms or the taxa that are being studied, but doesn't have all of the same parts and pieces. So if you want to think of it, you can think of this outgroup as being your comparison group. Okay, whereas the taxa that are being studied, these guys, would be called the in-group. All right, and this is what the final cladogram for these organisms would look like, and it's based on the traits in that table. So notice how every time you approached a branching point, they kind of put in why it was branching. So here, everyone from here on out is going to have a vertebral column. If you branched off before this line, it tells us that you don't. So no vertebral column, but everyone else has a vertebral column. Here, the branching point is going to be based on if you have a hinged jaw or not. So after this point, everyone has a hinged jaw. But if you diverge before that point, then you don't. So both of these, no hinged jaw. On this one, it would be the presence of walking legs. So everyone from this point onward would have walking legs, whereas these, if you diverged before that, you would have no walking legs. Then here it would be an amniotic egg, an egg that has a shell over it. So everyone from this point on has an amniotic egg of some sort, whereas if you branched off before, then you don't have a shelled egg. Then the next one would be here, I think. Yes, here. If you branched off after, you have here. If you branched off before, you don't have here. So that's kind of how the table is set up. So hopefully this wasn't too rough. Like I said, this is one of those topics that you learn and will understand a whole lot better after we've practiced and put some things into place and like moved some stuff around physically. But you know, these are just the terms and some of the information that I need you to have beforehand. So I hope you have a wonderful night, and I will see you guys soon. Bye-bye.